8, 9.
Hello. Hello.
No.
Hola. Hola, Joaquín. Estamos probando los micros. Rubén Calvo. Rubén Calvo Ibáñez. Que suba al escenario a recoger el premio a la mejor charla. Hola. Hola. Hola, hola. Pues ya está, ¿no?
¿Qué tal? ¿Cómo estamos? Sí, con el test. ¿Qué hacemos? ¿El HDMI? Sí. Va. ¿Dejamos este aquí o lo quitamos? Este. Él quita ahí.
Mira tú. Pues venga, el primero. Así.
Ahora.
Sí, sí.
No,
Hola. Uno, dos, probando. Good morning, everyone. Welcome to this morning session for this Wednesday of the Granada Seminars. I'm Luigi San. I will be your chairman this morning. And I'm super happy to introduce the man himself, Miguel Angel Muñoz, who's going to talk about microbial networks in health and disease. So thank you and welcome. Please.
Thank you. Is this working? Yes. So, good morning, buenos días. Thank you for being here after dinner and dancing and music last evening, a few hours ago, actually. and today I'm going to talk to you about microbes, essentially a few words we have produced, and also a lot of things that I learned from different sources, and I want to share with you because I think it's a very fascinating topic. I'm a newcomer. I've been working on this for, let's say, 3 years, not more than that. But I'm very, very fascinated, and I hope to share some of my fascination and this with you. Okay, so the first observation I want to share with you is that the Earth is supposed to be 4.2 billion years old, so it appears around here, the universe whatever started, and microbes appear on the surface of Earth very early. It's almost nothing here if you compare to eternity. Well, this is span, right? And very soon after, here you have the emergence of oxygen. So what happened is that these guys that appeared and started to take advantage of the energy that they found in very different places. Here it's a very unfriendly place for life because the temperature is extremely hot. It's Yellowstone, as you may have recognized. And you have these different colors correspond to different bacterial species operating in different conditions. Here in particular, this is a gradient of temperatures, okay? And and you see these many different colors spanning different conditions. So 2.3 billion years ago, we had the first cyanobacteria, which are these guys who were clever enough as to develop a technology to take advantage of the sun energy arriving to the earth, so making photosynthesis. Okay, so they were the first, and as a byproduct of photosynthesis, they start releasing something toxic called oxygen, and the oxygen formed an atmosphere, and after that, all the blooming of life that we know appeared. But just because microbes before constructed our atmosphere. And they are everywhere. They occupy every possible niche or source of energy on Earth, and they are essential for everything. For example, this is Serena, who's working now in the countryside, taking care of plants and trees. So for the soil to be alive, to work properly, you need a bunch of processes like fixing nitrogen from atmosphere, and this is done by some type of bacteria. Then degrading it to NH3 is another thing, nitrification, denitrification. There is a whole loop here, which is done by bacteria and other microorganisms, right? Also, and without these loops, nothing would work. So all the ecosystems as we know them rely on microbial loops of recycling of energy and products and substances. Same thing for the oceans, I don't enter details. And I want to quote this gentleman, Martin Blaser, who's the director of the Human Microbiome Project, and he said, they made the oxygen we breathe, the soils we till, the food webs that support our oceans. Slowly, inexorably, through the trial and error across the deepness of time, they invented the complex and robust feedback systems that to this day support all life on Earth. Okay? Feedback systems is something I want to underline. This is a selfie of my tongue. That of course is colonized. We are all colonized by bacteria, so we are a niche for them. And I like this one. So if this is on a scale, this is the number of cells we have, it's about 3 trillion. And this is the number of non-human cells we have in our body, which is the factor of about 2-3 times larger. Before they thought it was 10, now the number is 2. So we have much more cells in our body belonging to bacteria and other microorganisms than human cells. Of course, they are smaller, so, but anyway. But what is even more striking to me is this proportion here. After the Human Genome Project, we discovered we have about 23,000 genes in our body, each of us. Instead, we have 2 million bacterial genes. Every gene can do something, and seem do some specific function. So there is a factor of how much? 1,000? 1,000 more or 100 more genes that are alien to us. So as you can imagine, there are a lot of things that these guys are doing for us, living in our body, in our guts. So people, including this Dr. Kinross, proposed this analogy with physics, with dark matter. So in physics, we have normal matter is just 5%. And then they say we have this dark energy and dark matter, that it's essentially everything, and we don't know anything about them. So with biology, with genetics, it's pretty much the same. What we know with the Human Genome Project is a small percentage of the total amount of genes we have in our organisms. And what do these guys do in our gut, essentially, but all over the body, but especially in the gut? Well, here you have a list. You can pick up your favorites. Then, for example, let me focus on this. They produce many of the precursors that the body used to construct neurotransmitters, tryptophan for serotonin. This and the GABA, for example, is produced in the gut. Okay? So many neurological diseases or imbalances are produced at the origin because we have imbalances in the way these guys are processing the food we are giving them and producing something else. Okay. Of course, they also, for example, this one is especially important. They are crucial for the maturation and regulation of the immune system. So the immune system is exposed to them, and the immune system learn to distinguish what's self and what's not. So a lot of immune disease problems that we have in Western societies today are coming because we are very poorly exposed to microbes. Children do not play and eat soil as we did anymore, and we have an over cleaned atmosphere. So that's a problem for immune system and the development of allergies and all these kind of things. Okay, I want to more. If you want, this is very schematic. This is from a very recent paper, and you have a bunch of bacteria here and how they are interacting with each other, producing something and eating something else, creating these amazing networks where you also have to put on top of this the network of human cells, immune cell systems, and our own taking of the different projects. Here you have, for example, the GABA that I mentioned before, but many other things that are essential. Okay? So I would say that for sure this is the topic in actual medicine. Everybody, in all possible area of medicine, people are talking about the influence of microbiota, which is a microbiome, which is as we call this community of bacteria in health and disease. Again, you have here Martin Blazer, and what is becoming more and more clear is that many of our diseases in the modern world are associated to what we call dysbiosis. So an imbalance in the way microorganisms are established. And that's due to our diet, which is very poor, our lifestyle, our antibiotics. We are giving antibiotics everywhere, also to the things we eat. So this is altering, disturbing very profoundly the communities, and this is causing a lot of diseases. Of course, in each part of our body, we have a different community, and imbalances are specific to the community that we want to understand. Okay, this is a more radical example. People are doing fecal microbiome transplants, that you can imagine what that means, and the results are spectacular. So people are recovering from diseases like obesity, just changing the community that you have in your gut. Right? The most radical example that I like, and I mention it, is the infection with Clostridium difficile, which is a very nasty bacteria. So you get some infection, you go to the hospital, and they give you antibiotics, and the guy doesn't die, and they give you more antibiotics and even more antibiotics, and at the end, the community is absolutely dissolved. It's not there anymore. So what happens? What happens is that some opportunistic bacteria take over and colonize the whole thing as this Clostridium difficile, right? And then you get the diarrhea and nothing is working, and you essentially, in the worst cases, just die. Okay? So the only thing that's working in those desperate cases is fecal microbiome testing. So you go to a healthy person, take some time, and it's miraculous. The day after you go home, because you have recovered, you will create a new community immediately. It's a very radical approach that I don't recommend you to try at home. This is my personal motivation. I started reading at this because I myself has an autoimmune disease. So I start reading about the origin of this and how to modify, control, regulate the community of microbes. I learned a lot of things. So then privately in the coffee break, if you're interested, I can give you my own recipes that are my own. So I cannot say them here in public. I skip that. But of course, are well-informed recipes that I've taken from the literature and not from newspapers. Let's say. Again, this is for Serena. If you want to recover the fungi infection that you have in your garden, you just don't need to plant a new tree. You have to recover the whole community here, and you have to restore. You have to do microbiome engineering to recover the whole thing. Good luck. So, can we understand the nature of what's a healthy community, especially for autoimmune diseases? What's this biosis? This is a big question. How do you quantify, measure in an individual what's healthy and what's not? And of course, we are physicists, mathematicians, theoreticians. Can we make quantitative theory predictions about this? Okay, so our goal, this is for Eduardo Bataner, who is here, for physicists. We want to follow the steps of physics, of astrophysics. So first, we need to collect data. In those data, we want to look for empirical patterns, and then we want to look for theoretical principles. Is that possible? I start with measurements. So after the completion of Human Genome Project, we have a lot of technology, very advanced and beautiful technology to sequence DNA, RNA, so all the information. And people learn to apply that. Let me skip this. People learn to do that in a slightly different context, which is what this lady called metagenomics. Metagenomics means that instead of taking the DNA of a particular individual, what you do is that you go to an environment, let's say, for example, a lake or a sea, the ocean, you take a sample, and there you have billions of microorganisms. And what you sequence is the collective genome of this community, of course. And for that, they use, for 20-something years now, the term metagenomics. This gentleman here, who won the Nobel Prize, was a leading figure in, all these people are leading figures in microbiology. And this gentleman invented the term microbiome to refer to this collective genome of a community. Sometimes you will hear the term microbiome, and sometimes you will hear microbiota in Spanish. The difference is subtle. Usually microbiota is the set of individuals, species. Microbiome is the set of genes, but you can use them in this culture. And this gentleman, who's very famous, great inventor, sent this expedition, I recommend this book, to sample the oceans, to collect all the possible DNA on Earth. As they said, let's find all the proteins and genes that we have. Okay, let's collect them. So from those, that time started these big initiatives, the Human Microbiome Project, trying to understand all microbes in our body, and the Earth Microbiome Project to understand all microbes on Earth. Let me just mention something very briefly. When you look at the genetic material of these communities, you can do two different things. One is just to look for all genes. You detect sequences and detect genes. Okay, so you have the collective genes. But another thing that you can do is to identify the species you have. You can do that with different levels of resolution. And there is a very nice technology called 16S rRNA. I don't enter what that means, but what I want to stress is that you can identify species. Not only species, but all the phylogenetic tree. It's like you can identify. So when you look at data of these communities, the data looks something like this. These are the 16 main geni, like, let's say, family-like 16 main that we have in our body, the humans. Okay, and what you see when you look at some of these data is that you have bacteroides, bifidobacterium, I don't know, in different proportions. This is some healthy guys, and this is some unhealthy people, and you see that there is a lot of variability. Also, in some occasions, you can have the time curses. So for a given individual, you have how a given species changes across time. Okay, so now we are searching for empirical laws, and let me remind you or tell you that there is this science which is called macroecology. That if you look at the definition of what macroecology is, it's statistical mechanics, because they find statistical patterns in different large communities, propose and validate mechanistic models. So it's the collective things when you have many individuals in an ecological community and you look for patterns and laws, which are very universal for some species. They're amazingly universal, as you would say. So a lot of papers about macroecology and microbiology. This is a very fancy thing. It's been so for 2 decades. And here is my friend and collaborator, Jacopo Grilli. He's a brilliant guy. And he wrote this very influential paper in Nature Communications already 4 years ago, I think. So he took data from different samples from different communities and analyzed this time series and found 3 empirical laws which are amazing, in my opinion. So this one is the fluctuations of a given species. So, and you put the mean here at around 1, so you normalize this species to be centered around 1, and you put the fluctuations here, and you see that it's a gamma distribution. It's very well fitted. And the mean and variance, the variance is also adjusted, are not independent. You have a law relating the mean with the variance, which is Taylor's law. It appears already in this conference in some context. And you have the mean abundances is distributed as a log norm. And here you have like 12 different microbiomes with thousands of species collapsing in these curves that are very appealing to us physicists as meaning something, right? So these are called now the first, second, and third law of macroecology. Okay, and as I was saying before, you can have 2 different types of data: transversal, meaning that you look at the microbiome of different people, or of the same guy along across time, right? For both of them, you have the same laws. So talking now about mathematical models, it's like the ideal gas. That's what's the ideal gas for the hydrogen molecule, the easy model, if you want, approach to this. And the answer is this equation. The logistic growth is the most basic equation that you can imagine in ecology. It has a long history that we don't discuss here. So the abundance of species is just growing at some rate and then saturating when it reaches some carrying capacities and maximum value, right? Okay, and the new ingredient is that we introduce what's called environmental multiplicative noise, environmental fluctuations, meaning that the growth rate, the linear term, is changing in time depending on whether you have a lot of glucose, you have eaten a lot of the day, or very little, or whatever. So it's a random source. So if you take this simple model, what you find is that the steady state distribution is a gamma law, and with very simple assumptions, you recover the second and third law. So you can assume that species are independent because I didn't assume any relation between them. And each of them just sees like an average of the environment, which is fluctuating. And this average of the environment generates by itself the gamma distribution and the relation between the mean and the variance because of the multiplicative process. And then with some additions, you get also a third law. Okay? So these are the empirical laws for 8 different microbiomes. These are the same laws for the stochastic logistic model. So pretty good. But we have to go beyond the ideal gas approximation. We want to do something more sophisticated. This is just to illustrate. It's a recent paper, but already with hundreds of citations, in which people are studying the interactions between 2 species. So what they do is go to the lab and put in competition 2 different species with different abundances at the beginning and see what happens. Many times, like here, one of them dominates and the other one distinguishes. In some other cases, like in this column, you have more fancy fancier behavior with different things. So from this, you can have, here you have 10 species or 12, and you infer the pairwise interactions between them, and you can construct a network, and they claim with the network they can capture more than pairwise. So collective things are essentially captured by pairwise interactions, and higher order are not that important in this context, and some contexts they say. But okay, this is debatable. And here I just illustrate what I said before. Here you have 12 species, and here you have some metabolite that they produce, and here the other way around. You have some species, and the metabolite they eat, or well, this is they use, and this secret. So, and you can see that there are many things here in common, so that's why you have this interaction here. Because if you secrete something that it's eaten by someone else, there is a positive interacting cooperative link here. If two guys are competing for the same thing to eat, you are going to have a negative interaction coming out of competition. So the network of competition and of cooperation. So we went to the data. We started collaborating with Jacopo Grilli. We went to look at data and ask whether there are correlations. So this is some of the microbiomes, and this is a histogram of pairwise correlations. The gray line is what you expect just by random because of stochastic noise, sampling noise, and what you get is that you have a much broader thing, so there are a lot of correlations. Right? And we did our first contribution that was trying to answer this. And I will be very brief because I want to tell you a different story, but just let me give you the flavor of this first story we produced. What we wanted to study is how these correlations between species depend on phylogenetic distance. So for example, these two guys here are very close phylogenetically because they branch very recently from the same thing. Instead, for example, this and that are very far phylogenetically speaking, okay? This is like a tiger and a panther, and this is like a human and a mosquito, I don't know, okay? So you can reconstruct from genetics the phylogenetic tree, the phylogenetic distance, and study how the correlation depends on that. So in few words, we wanted to understand if things that are correlated is because they are similar and they do essentially the same thing, or not. Similar things are anti-correlated because they are eating the same, so they should be in competitive exclusion one with the other. So what's your guess? If this is phylogenetic distance, how should be the pattern? So this would mean competition, so similar things are negatively correlated. This would be the opposite, so similar things favor of the same environment and they go together, and this is flat, you have an idea. We don't know, so we look at the data and we found an empirical pattern, so it's my empirical pattern ever. I'm very happy, very proud of it. I didn't do it myself, but I'm very proud of this pattern. So what we have is that there is this type of dependence. So that very similar species are correlated together. And that means, we also have a mathematical expression, this is for 8 different microbiomes, a glacier, gut, lake, ocean, whatever, and they all follow the same logics. And our point was to construct mechanistic models. So let's take the stochastic logistic model and improve it. To improve it, you can do follow two different strategies. First strategy is go in the direction of log table terra models. Log table terra models are models in which you describe every species, bacterial species I, okay, and you have it's similar to stochastic logistics, but now you have this interaction term in which the dynamics of I depends on the population J, and you have some coupling, and depending whether it's positive or negative, you can have competition, cooperation, whatever, right? So this is direct approach, implementing interactions between species. Instead, you can do something a little bit more elaborate, which is what's called consumer resource of MacArthur's models, in which you have dynamics for bacteria, but also dynamics for resources. So here you are implementing explicitly that the interaction between bacteria is mediated by resources, by substances, by what they are eating and what they are secreting, right? So it's a more elaborate model, but in our case, we thought it was more convenient to have more details to model actual matter. I will skip absolutely this consumer resource model we produced because I want to go to something else. But just let me tell you that oops, this final line is the result of our model. These guys are the empirical data, and the key thing is that these interactions are dominated by what's called environmental filtering, which means that mostly they depend on how the environment fluctuates. If suddenly we eat a lot of iron, then all iron prone to it bacteria will proliferate, and it gives you this. Okay, so this is the first pattern, and we have some explanation for it. But let me just show you that this is the average curve, and that at every phylogenetic distance, like for example here, you have both positive and negative correlations. Okay, so this is not the end of the story. This is just a pattern. We explain it, but as I said, the pattern of correlations includes a lot of negative correlations and a lot of positive ones. Okay. So the next question is the following. Let's look now at simplified level. Let's look at the log-table terra level, just looking at the direct interactions between species. Can we infer the matrix A matrix from actual data? This is something what Mario Castro gave a fantastic talk on Monday. And the thing is that this is impossible. You cannot solve this problem for many different reasons. In particular, if you measure the correlations, you have n times n minus 1 values, and this matrix has n squared. So you're missing information somewhere, or n minus 1 half. And it's undetermined. So this is the second contribution in collaboration with people in Madrid, especially the group of Jose Cuesta. And what we did is an inference, Bayesian inference approach, in which we consider some initial metrics, and we look at the likelihood that this matrix generates the patterns that we observe, which are the three ecological, macroecological patterns of Grilli, plus our correlations between species that we have measured. Okay, so let's say this is the gamma, this is the Taylor law, this is the distribution of log normal, and this is the distribution of pairwise correlations. So we impose that these four patterns are preserved. And what we do is an optimization algorithm implemented as a Monte Carlo. This is a detail, it's not so important. So essentially we start changing the matrix to look for good matrices reproducing the patterns. This is the constraint, the optimization constraint. But importantly, what we are interested is in an ensemble. So we are not hoping to recover every specific interaction. That's hopeless, as I said, but we want to understand the statistical patterns of these matrices, of this ensemble of matrices. Which statistical properties do these matrices need to obey for the statistical patterns of abundances to be obeyed, right? So we did that, and the model is working, and the inference mechanism is working very fine. So we reproduce the three laws, and we reproduce the distribution of correlations. So this one is data, and this is the result of the inference matrix, one of the matrices in the ensemble. So we are very, very happy. So what are the properties of this network, this ensemble of networks of interactions between microbes? Well, the main thing is that they are very sparse. This is the main observation we made. So during the optimization process, many of the variables of the interactions went quickly to zero. This is an interesting observation and has a lot of relevance for what Mario was describing the other day, what are called relevant variables. So, I mean, we can discuss about the meaning of that, but at the end of the day, the result is that the networks are very sparse. Okay, but I was very disappointed, personally, I have to confess you, because I was expecting these networks to have some fancy topological properties, distinguishing them from random networks. But unluckily, or luckily, I don't know, they don't. So here I am comparing our networks with just random networks with the same connectivity. So this is the degree of mutualism. Mutualism means that the interaction ij is positive, the ji is also positive. So the two guys are helping each other, and the different ecological communities you can interactions you can classify the piece. This is for the model, and this is for random. It's the same. This is the in-degree distribution of connections. Out degree, same thing. Number of loops of different lengths is the same, random and Monte Carlo. This is the spectrum of the matrix. Again, that is identical, no big difference. So I was kind of disappointed because the networks cannot be random, and they are sparse, but cannot be random because random matrices do not reproduce the empirical patterns. So we are still missing something. Is that we have not yet looked at the right statistical patterns of the networks. But then one day I came across this paper. It's by Jordi Bascomte, collaborator of Enrique, who gave us a fantastic talk about the evolution of life. And in this paper by Jordi, the group of Jordi, they discovered, looking at other ecosystems with plants or whatever, what they discovered is that when you have this type of interaction motifs, so this species is in competition with this and with this, but these two are helping each other, or this other one. So essentially, these are frustrated triangles. Whenever you have these frustrated triangles, biodiversity increases. So this is the abundance of this motif, and the more you have, the more biodiverse is the ecosystem. Same thing here and here. So I said, oh, maybe our microbiome networks also obey this. Maybe they try, because we just look the number of loops, but this is not just loop, this is a motif. You have to look the statistics of these motifs in the network, right? So we want to look for this, and this is work in progress. Ruben here in the audience is working deeply and very carefully on this problem, but the preliminary result is that yes, it's true. In our microbiome sets, we have much more of these triangles than in random networks. So our A networks, inferred network are not random, and we think we're learning why, because with these things, you improve biodiversity. For example, just to explain you one, if this is a guy eating this and eating this, if the two of them help each other, they will not get stung, right? So it's a way to survive the predation of one. And you can make similar reason for this. So this type of structure favor biodiversity, and they are present in our networks, and they are not present, so overrepresented in random networks. So in the last, 10 minutes or so of the talk, I want to tell you about our most important, relevant, and exciting contribution, and very happy with it so far. And it's trying to apply all these general concepts that I talked to you about, which are for any generic microbiome, soil, gut, whatever, lake. Now I want to focus on the human gut, on health. Can we distinguish a community from a healthy individual from an unhealthy individual? Some people, many people, everybody's trying to find this. This is like the Saint Grail of the field. Everybody's trying to understand this. This is a 10 years old paper in which they claim, for example, that all human gut microbiome can be classified in 3 groups that they call enterotypes. So you either fall here, here, or here. But now it's not, people don't believe that anymore. Some other people are talking about alternative stable states. So it's like you can have different type of stable symbols of communities. Some of them are healthy, some are not. This, of course, needs to have some truth. Some people are saying that the states of dysbiosis correspond to reduced biodiversity. And this is kind of true. Here you have different diseases. For example, let's look at Clostridium difficile that I mentioned before, CDI, where is CDI here. And you see that in people with this infection, the biodiversity is way lower than in healthy individuals, right? And that's easy to understand. Also in people with inflammatory bowel disease, all these digestive disease, you also see it, but not systematically. This is, for example, people with colorectal cancer, and here it's not working, right? So reduced biodiversity is okay, but it's not the full story. So we still need markers of dysbiosis. Can we do that? Okay. So this is part of the thesis of Roberto Corral, and he did an amazing work. He has been working on these simulations that I will present in 3 minutes for 2 years. So let's give it 3 minutes, one for each year. So the model that Roberto implemented is a model of consumer resources. So we have here the dynamics of species abundances, bacterial, right? And we have the substances that we call. And these are things that can be eaten or secreted by different species. And the model looks a bit sophisticated, but it's pretty simple. Just let me guide you a little bit through these equations very briefly. So for example, each species is eating some substances here with some growth function, it's here, but it can do it. I mean, this matrix P tells you what this species I is able to do, is able to eat beta and secrete alpha. So it's a metabolic pathway from substance beta to substance alpha. And this matrix specifies the identity of this species. This is what this species is able to do, right? So the model is getting more sophisticated. Now we have to implement personality to every species. And this is the energy that it will get to grow, just transforming substance beta into alpha. And here is the same thing, essentially, for substances. So they get added, so there is an inflow of substances. They get diluted because we go to the toilet every day, and they get eaten because of consumption, and they get also generated. This is important. This is called cross-feeding. Is the fact that substances that are secreted by some species are eaten, can be eaten for another one. So this is a positive term. Some substances are growing because are generated by it. Okay. The other ingredient is that the substances are ordered in a gradient of energy. So you can degrade from here to here, from here to here, or give big jumps, but you cannot take this very little energetic substance and generate this one, of course. Okay. So essentially, for example, this can be glucose, and from this you generate many, right? So we put this in a computer. The identities of bacteria are generated randomly. So these matrices are random. This distribution here is random. Everything is kind of random, but as it's running, it generates spontaneously some type of dynamics. So you have time series, and we check for the statistical patterns. And I was amazed squared, or amazed cube, when I saw that without doing anything, just looking at the data, the statistical patterns of Greeley's laws were there. So the distribution of the species is gamma. We have Taylor's law, and we have that the means are distributed as a log norm without doing anything. Okay? No noise, and not even that. There are more statistical patterns. I showed you a lot of macroecological papers, and they have some patterns that are here, and all of them are reproduced by our data without doing anything. So our model is generating something that resembles, that resembles the dynamics of real microbiomes. For example, one property of real microbiomes, and this is for Herbert Levine, who asked me about this, you see a lot of variability. So if you look at the time series and you focus on one specific species, you see that it's changing very wildly in our model and in reality. But if you do a little bit of coarse graining, and instead of looking species, you look for classes, it's very stable. And if you look for functions, meaning, for example, the substances you have in the community, it's very stable. So this is one of the main properties of here. One of the main properties of the microbiome, human microbiome, is that it's very volatile at the level of a species, but very robust at the level of functions, what's actually doing. Okay? So you may have many different ways to do something, but that's something that's very stable a long time. And that appears spontaneously also in our model. And this is a funny observation. We realize, Roberto realized, looking at his data, that the simulations usually look very chaotic, so everything is changing a lot. But suddenly, from time to time, not so often, but from time to time, you see these collapses, we call it collapse, in which the community gets like to a fixed point, it gets trapped with some fixed abundances, and then at some point also spontaneously it escapes, because we have random addition of species and substances, so at some point it escapes. But we have these two different types of situations. We call this the collapse situation and the fluctuant situation. This is, for example, the Shannon entropy. Here is much lower and here is larger. The number of enzymes, of products that you have, here is much poorer. When you have this collapse state, it's extremely poor, right? So it seems like a reduced diversity situation. And while this is also to illustrate in the health in the fluctuating state, we have a lot of pathways active from some substance to substance that actually they are happening. In the dysbiotic, this much reduced. Okay, let's skip this. So this is our serendipitous observation, and I will finish in 3 minutes, which is the following. We wanted to characterize mathematically what's the difference between these two possible states. Okay? And the thing is that as we have the model and it's running in the computer, we can look at every particular moment, what are the interactions, how are the interactions. So for example, if two bacteria are eating the same thing, we quantify how much amount of that thing we have, and we can measure in the equations what's the level of competition. Or what's the level of cooperation, seeing how much one is producing of some product which is eaten by them. So we measure the degree of interactions in our model. And what we see is this thing here. This is the fluctuating state, and this is the balance of positive minus negative interactions. So in the fluctuating, it's essentially negative. So there is a lot of competition. It's like a capitalist society. Everything is competing, and the result is very, very fluctuating. And that seems to correspond in our proposal to the healthy state. Don't take political implications of my talk, please. And instead, in the collapse state, is dominated by cooperation. That's very disturbing. I wanted just the opposite to happen, but it's not the case. What happens is that in the collapse state, there are some consortia of microorganisms, think for example of Clostridium difficile, that essentially is taking up all the energy and producing some very residual substances that are eaten by a few other species, and that's it. So the whole structure of the community is removed, and you have a strong cooperation between a few species, right? This is a detail, the composition of the person. It's the same thing. So now the final step is, okay, so let's look to real data of patients and healthy people, infer networks from those data. This is tricky, but we did it with 3 state-of-the-art algorithms with different technologies that I will not discuss here. It is not our own work, but I just want to show you the results, which is extremely robust. Here you have, this is a fraction subset. So instead of using the whole data, we use 90% or 30%, right? Just to be sure that our results are consistent, this kind of bootstrapping thing. And these are people with inflammatory bowel syndrome. And these are healthy people in the same data sets. So all healthy people had this indicator, which is essentially how many positive minus negative interactions you have. So you have much more positive cooperative interactions in unhealthy people. So dysbiosis is always characterized by a positive value of this indicator we have invented. And this occurs across different, this is for species, this is for genii, and this is for family. So on many different taxonomic levels, you have the same effect. And I don't show you more evidence. Well, here is in the model, and here's for inflammatory bowel disease, inflammatory bowel syndrome, Clostridium difficile. For all diseases, autoimmune diseases, we found there is this imbalance. It's an observation that made us associate the presence of dysbiosis to an imbalance in the signs of interactions. We change from competition-dominated healthy states to cooperative-dominated or cross-feeding-dominated communities in unhealthy states. Can we use this as an early warning of disease? That would be great, right? And the answer is we don't know, but we have this thing, which is the following. On the top, you have model data fluctuating. This is the balance in fluctuation and much higher value in the collapse. And in the collapse, as when you enter the collapse, as time goes, you go up. So it's every time more collapse. And these here are data for patients with colorectal cancer, in which they made sampling of the microbiome at different times. And at the beginning, so we took this data and we measure our index, and the index is growing. Yeah, I was about to say nicely, but I don't know if it's uglily. Is growing across time. So as the disease gets worse, our index is increasing. So we are proposing this to medical clinical doctors as an indicator to detect in advance. Yes, this is how our indicator correlates with biodiversity. And well, I skip it. So the take-home messages are: I hope I convince you that microbial communities are ubiquitous and essential for life. They are crucial. They are not alone. They interact with each other and they form very complex networks with non-trivial features which we are just starting to grasp, to try to understand. We have a new insight on what's dysbiosis, how the networks change when you have one of these diseases, what's the essential thing, this imbalance. And I hope I convince you that this is a very exciting, relevant, fascinating topic for quantitative science. So I have to thank all my collaborators. The first paper I mentioned is with Jacopo and Matteo. The second is with a group in Madrid, including also again Matteo. And the third one is with Roberto and my ex-PhD, now professor in Rutgers, Juan Monachela. But we joined forces to take this forward with Michael Manhart, also Simon Levin in Princeton, and especially this couple, Martin Blazer and Gloria Dominguez, who are the chiefs of the Human Microbiome Project, and they were convinced by our arguments, so they signed the paper, and luckily it will get in science. We are epsilon distance from being accepted, but not accepted yet, so maybe I don't mention this. So thank you very much for the attention, and ready if there is time for questions.
Thank you, Miguel Angel. Beautiful and inspiring. I have a question about these two states that you've mentioned, the fluctuating and this collapse state. In the simulation, I didn't get the model well. I can ask you later, but in the simulation, it looks like there's some sort of intermittency. So you go from one state to the other and then return back. Do you expect that in patients, for instance? So did this return to the fluctuating state?
Yeah,
It always goes back, or do you have to do something to recover?
that's a very good question. So essentially, in the simulations, you can run forever. And for example, this, if you take to real time, it would be like 50 years. Okay, so if you wait enough, essentially there are instabilities, and you eat a whole set of new bacteria. These things can happen spontaneously, but depends on your diet and depends on the structure of it. And the times are difficult to compare. So the short answer is yes, but the time scales can be unrealistic for us.
I guess I have one technical question and one more philosophical question. The technical question, I guess, is also related to this. How do you take into account either approximately or demographic noise? In other words, the fact that the population really gets small enough, some species will go extinct and won't be able to recover.
Yeah,
So
whenever they go in the
you
model.
you put it directly a cutoff in your model.
Yeah, the model is deterministic, but whenever a species goes below some threshold, which is 0.01, we distinguish it. So there is demographic noise.
Okay, that was the technical question. The general question is, it seems reasonable maybe that the dysbiosis could be a marker of disease, but how are you going to explain let's say, the onset of disease without coupling it to the immune system in some way, which after all is the cause originally
Sure.
of inflammatory bowel disease
Sure.
and some of the other
Sure.
symptoms you referred to.
Absolutely. So we have just half of the story. I mean, half. I hope we would have half of the story. We have the microbiome part, and then we have the interaction with the host and the immune system. We are clueless about that. So what we are saying is just by looking at the microbiome, because of imbalances, bad diet, bad habits, antibiotics, you can get into these collapse states. Then there is a lack of metabolites, and that can interfere. So that's all we are saying. Then the second part, of course, is absolutely to explore how that interact, or what's the genetic predisposition of the host for these things to happen or not to happen, or to remediate them, to cure them if they happen. All this is, of course, to be explored.
I think we can have time for two more questions, but if I can ask the next speaker, if I can ask the next speaker to come forward, please.
So thank you very much for the talk. I think it's illuminating, very nice. I missed that in your explanation, but when you mentioned the higher order interactions, do you have a kind of mechanistic idea of why they emerge in this situation?
I would say if you just assemble a community randomly, with interactions randomly, and then you let it go, if you do not have these type of things, it would go to extinction, or you would lose species. And the more of these guys you have, the more species you will have. So maybe, I'm including, I'm just making it up, but probably just this selection process, that this type of interactions favor biodiversity, and so that's why you find them in real communities. If you didn't have these, the community would be extinct, something like that, and a posteriori explanation.
Very nice talk. Regarding the second law, you have a Taylor law.
Yes.
The exponent is...
It's very close to 2, but
so
we don't make, but it changes depending on community 2, 2.1, 2.3.
This could be very interesting. So it's 2, it will follow also the generalized Taylor law.
Yeah.
But it could be possible exactly, in some communities there are deviations from this exponent, and there are...
Yeah, the thing is that data, of course, are very noisy. So between 2 and 2.3, 2.4, it's difficult to
But you don't reach 2.6 or something like that.
I don't think so.
Okay.
Okay.
1.6. As low as 1.6. Okay. I don't remember.
Okay, I think this is all the time we have now, because we have the next speaker here. So thank you very much once again, Miguel.
Thank you.
Sí, aquí está, mira. Aquí está.
Okay, so after this very nice talk, we have the next speaker, Laia Barjuan, who is in Barcelona. In what center? At the University of Barcelona. All right. And she will be talking about the multi-scale self-similarity of the weighted human-brain connectome. So we go from biotome to the brain once again. It is working. So the stage is yours.
Thank you. Do you hear me?
Yes. It's not working.
No, sí, sí, funciona. O sea, es que funciona. Sí, sí que pasa, que funciona. Um, okay, thank you. My name is Laia Barjuan. I'm a PhD student at the University of Barcelona. And today I'm going to talk about my latest work. But first, let me start my talk by asking a question to all of you. What do... Sorry. Es que me suena.
No.
Vale. Okay, yeah. Ahora sí que funciona así. Sorry for the technical problem. As I was saying, I'm a PhD student at the University of Barcelona, and today I'm going to present my latest work on cell similarity in the brain. But first, let me ask you a question to all of you. What do Romanesco broccoli, snowflakes, and river networks have in common? As I just said, it's self-similarity. Self-similarity is surprisingly common in nature, and it consists of patterns that repeat across scales, shapes that look the same whether you zoom in or out. We find it everywhere in the structure of the trees, in the branching of the trees, sorry, in lightning bolts, and even within the human body, in the structure of the lungs, or in the complex network of blood vessels. And so we wondered, what about the brain? Is it very similar? This is the fundamental question that we wanted to ask in the work that I'm going to present today. We didn't look at the shape of the brain, but at the connectome, which is the network of anatomical connections between different brain regions. This work was done with my collaborator Mukwa Zeng and my supervisor Maria Ángeles Serrano. To study that, the first thing we need is a multi-scale unfolding of brain networks. And to obtain them, what we do is divide the total volume of the cortical brain into different number of brain regions in the interval from around 60 to 1,000, specifically in the number of nodes that I display here on the slide. These regions will be the nodes in our network. We will connect two regions when there are nerve fibers linking them, and we assign to each connection the density of nerve fibers as a weight. From now on, I will refer to the networks with 1,000, 400, 200, and so on as layers 0, 1, 2, 3, and 4, respectively, of the multi-scale unfolding of networks. We worked with two independent data sets. The first one had 40 subjects aged between 20 and 30 years old, and the second one had 44 subjects aged between 22 to 35 years old. In the first one, we had 16 females, and in the second one, 31. And I must remark that this work was done on healthy subjects. In previous work from the people in my group in Barcelona, it was already shown that when regarding binary connections, which means that we just look at whether a connection exists or not, and ignore the weight, the brain exhibits self-similarity. The topological properties of the brain are self-similar. This means that properties like the degree distribution or clustering look alike independently of the resolution that we are observing. But two brain regions may be connected by lots of nerve fibers, whereas some others may be connected by just a few. And we cannot treat these two connections as if they were equal. So we ask the following question. Do connection weights alter the observed cell similarity in binary connectomes? When we study the weighted topological properties of multi-scale connectomes, this is what we find. In here, I will show the results for layer 0 in red, the one for layer 1 in blue, and so on, as you can read in the legend. If we look at the distribution of weights, we see that the curves corresponding to the different layers collapse under the same curve almost perfectly. And therefore, we can see that the distribution of weights are self-similar. In addition, if we analyze each independent curve, we can extract other valuable information. For instance, in here we see that the weights span over more than 4 orders of magnitude, with values ranging from 10 to the minus 5 to 10 to the minus 1. And therefore, they are very heterogeneous. Moving on to the distribution of strengths, and we define the strength of a node as the sum of the connection weights, of the weights of the connections attached to it. And again, the self-similarity is remarkable. If we now study the relationship between strength and degree, again, the points corresponding to the different layers collapse under the same area of the plot. We can see that the relationship between the strength and degree is a power law, and the strength is proportional to the degree up to a certain power, which, interestingly and contrary to the behavior in other real-world networks, it's close but smaller than 1, which means that high-degree nodes tend to have less strength than what would be expected given their connectivity. And finally, the disparity, which measures the heterogeneity in the weights of the connections around the node. Again, the points corresponding to the different layers overlap in the same region of the plot. Also, this region where they overlap is between the completely heterogeneous case, which would be the brown diagonal, and the completely homogeneous one, which is the x-axis. Therefore, we can say that the weights are heterogeneously distributed in the neighborhood of a node, not completely heterogeneously distributed, which would mean that just one connection accumulates all weights, but all strength, but they are not homogeneous. Nice. Given these results, we can say that the weighted topological properties of multi-scale connectomes are self-similar. But the weights of the connections can give us much more information than just that. Let me change of topic now for a bit, and let's each of you think about your social network. Think about your closest friends, your family, the people you talk to every day. And then think about your neighbors, your old friends, the former classmates, your acquaintances, people you only occasionally interact with. In network terms, we would say that the connection between you and your close friends is stronger than the connection between you and your acquaintances. So then we can ask, which connections are more important, the weak or strong ones? Our instinct might be to answer the strong ones, and in many ways that makes sense. If you need advice or support, someone to talk to regularly, you will turn to your close friends. But now imagine that you lose your job and you're looking for a new one. Your friends and your family live in the same social circle as you do. They hear the same news. So where does something new come from? It's usually the people you barely know, the neighbor, the old friend, the ones that connect you to different information, to a new opportunity. And that's what sociologist Mark Granovetter discovered some decades ago as he postulated the weak ties hypothesis. And it can also be applied to the brain. In neuroscience, however, the weights of the connections, especially the weights of the structural networks, have often been overlooked. And when they haven't, weak connections have usually been considered negligible. But recent research has shown some evidence that the weak connections in the brain tend to link different communities too. Then we wanted to explore how are weak connections organized in the brain. We checked the occurrence of the weak ties hypothesis by measuring the tendency of weak connections to concentrate across modules, understood as groups of densely interconnected nodes. We used the Lowen algorithm for community detection in unweighted networks to detect these communities, and ignoring the weights of the connections in this community detection process ensures that the obtained partition is not directly influenced by weights. The easiest thing we can do if we want to check if the weakest connections are the ones that tend to link nodes in different communities is calculating the average weight of intra-community connections, which are the ones that link nodes in the same community, and the average weight of inter-community connections, which are the connections that link nodes in different communities. If we calculate this is what we find. In here, the average weight of inter-community connections is shown in orange, and the average weight for inter-community connections is shown in blue. We have error bars for inter-community connections because we calculated the mean weight in each community and then averaged over all the communities in the network. And as we can see in this plot, inter-community connections tend to have lower weight than intra-community connections. So this is great. This is what we wanted, but it is a very general measure. And we wanted to be a little bit more precise. To do so, what we did was to construct a nested hierarchy of filtered connectons with a given percentage of the weakest connections. So for instance, in here, the network in orange would be the original one, and then the network in purple would be the one where I have kept only the 20% of weakest connections. As you can see, there are the thickest connections in the original layers have been removed because I display here the thickness as a proxy for weight. We did this for 1%, 10%, 20%, and so on. And then we calculated the density of intermodular connections in each filtered connector, which we denote as RE and N, where N is the percentage of edges remaining in the network. The density of intermodular connections can be calculated as the number of intermodular edges divided by the total number of edges in the network, which in the purple case would be 1, 2, 3 divided by 5. And finally, we normalize this measure to be able to compare across original layers by the density of intermodular connections when all connections have been considered. So by RE 100, sorry. And we call this measure the normalized density of intermodular connections. If I display the value of the normalized density of intermodular connections versus the percentage of edges remaining in the network, we see that this measure has an exponential decrease with the percentage of edges in the network, remaining in the network. And it is an exponential because the x-axis is in log scale. So for instance, when we are only keeping 1% of the weakest connections, the normalized density of intermodular connections almost, well, more than doubles the value that we get when considering all connections. And that happens across all scales. Okay. So, given these results and the ones that I presented in the previous slide, it is clear, or it is fair to say that experimental weighted multi-scale connectons are self-similar, and the weakest connections are the ones that tend to link the nodes in different communities. We have just observed that, but we are physicists, we want to model things, and we wondered if we could reproduce these results using a suitable model and denormalization techniques. As they explained yesterday, renormalization allows us to zoom out while preserving the essential features of the system. It helps us identify invariant properties across scales and reveal universal patterns that transcend the details of the individual components. In the context of networks, there are multiple ways to perform renormalization, but I will focus on geometric renormalization. which involves systematically cross-graining a network while preserving its underlying geometry. Geometric normalization is based on the S1 model, in which each node is characterized by two variables: the hidden degree kappa, which measures its popularity, and an angular position in the similarity space, that is the one-dimensional sphere or circle that we use in this kind of model. Then the connection probability in this between two nodes in this model, in the one-dimensional version of the model, is given by this equation here, in which d is the similarity distance and can be calculated as the radius of the similarity space times the angular separation of the nodes. Then mu controls the average degree, beta controls the average clustering, and as I said, kappa I and kappa j are the popularities of nodes I and j and are proportional to the degrees of nodes I and j, respectively. One interesting thing about this model is that the name similarity from the similarity space comes from the fact that this coordinate captures the affinity between nodes. So when two nodes are more similar, they are closer in the similarity space and therefore more likely to form connections. Then the coordinates or the angular positions in the similarity space offers information about the community structure in the network. So that if the model is a good fit for our data, we would expect angular coordinates to correlate with neuroanatomical information. So that nodes belonging to the same neuroanatomical region are concentrated across a narrow section, angular section of the similarity space. This is the model, and the first thing that we need to be able to perform geometric normalization is an embedding, which means that we need to find the coordinates kappa and theta for each node that maximize the likelihood of the network being produced by the model. We did this with Mercator, which is a model-based tool to map networks into the hyperbolic disk that was developed by the people in my group some years ago. And before explaining how the geometric normalization procedure works, let me show you what an embedding done with Mercator looks like. This is not from this work, it's in fact from the previous work that I talked about self-similarity in binary connectons, but I thought it would be interesting to show it here. So, as you will have noticed by now, in here they were not using the S1 model because the nodes are not placed along the similarity circle, but they have a radial coordinate. This model is equivalent to the S1 model. We just use it because it's better for visualization purposes. And in here, we transform the kappa coordinate into a radial coordinate. In here, they colored every node by the neuroanatomical region that it belongs to. So I know it's very difficult to see it with the naked eye because there are a lot of colors, but we will agree that the embedding is able to group together the nodes that have similar colors. In fact, if we show the neuroanatomical regions in the place where the nodes belonging to it are located, this is what we find. And in addition, we can also paint, or we can also show to which hemisphere each node belongs to in a different color, and this is what we find. So Mercator and the embedding done with Mercator naturally separates almost, I will not say perfectly, but it separates very good the two hemispheres in the brain. Now that we know what an embedding done with Mercator looks like, I can move on to explain you the geometric normalization procedure. Let's go back to the S1 formulation because it will make things easier. Once we have the embedding of the highest resolution layer of the connectome, we proceed in several steps. First, we define non-overlapping blocks of consecutive nodes in the similarity space, in our case of two nodes. Then we cross-grain the nodes in each block to form a supernode, and we assign an angular coordinate within to the supernodes within the angular region defined by the nodes in the block. In this way, we keep the angular ordering in the original connector. Then we connect two supernodes if there is at least one connection between a node in block I and a node in block J. So for instance, in here, between these two blocks, we have these two connections, and therefore we need to establish a connection between the two supernodes. Whereas in these four nodes in the constituent layer, we don't have any connection between them, and therefore we cannot establish a connection in the coarse-grained layer. To assign the weights to each connection, what we will do is keep the maximum among the possible weights among the possible weights of the connections that we have in the constituent layer. So for instance, as I said, between these four nodes, there are two connections, let's say with weights 0.2 and 0.5 respectively. So we would keep the connection with weight 0.5 in the coarse-grained layer. We can do this procedure iteratively until we have as many coarse-grained layers as we want. This is what we did. We performed the geometric normalization 4 times, starting from layer 0 in the original connectome, and we calculated the weighted topological properties that we had studied before. And this is what we find. In here, the results for experimental connectomes are displayed with the markers, and the results for the model are shown in the solid line. As we can see, the agreement between the results, the experimental results, and the ones predicted by the model is very good. I should say that here we had to rescale the weights and strengths because given the nature of the geometric normalization procedure, in which we increase or we keep the maximum weight among the ones possible, the weight and the strength increases as we coarse grain the layers. And finally, we also calculated the results for the weak ties. And again, the original trend is replicated. Maybe we could say that the agreement between the coarse-grained layer 4 and the original one is not very good, but that's because it is the layer that is most affected by finite size. Then we have shown that the weighted geometric normalization procedure is able to reproduce both the observed self-similarity in the weighted topological properties of experimental multi-scale connectomes and the organization of weak and strong links at all scales. To sum up, the take-home messages from our work are the following. First, we have shown that the weighted topological properties of the multi-scale connectomes that we are studying are self-similar across the 5 resolution scales that we examined. Then we have shown that the connections in the brain organize according to the weak ties hypothesis, with weak connections linking nodes in different communities. And finally, we have demonstrated that the weighted geometric normalization flow effectively reproduces both the observed self-similarity in the multi-scale connectomes and the organization of weak ties at all scales. And this is all. Thank you very much. Here you have the theoretical of our paper, and I will be glad to answer your questions.
Thank you very much for a very beautiful talk. We have some time for questions? So, if not, I do have questions. We have one here.
This was a nice talk. I was curious about if here all the data, the weights are positive in this context, right? I wonder if you have thought about the possibility of having negative weights. I know this is not common in this kind of data, but in other scales or kind of experiments you can find that. And I wonder if this normalization procedure, how it would be modified by having like side coordination weights.
Yeah, great. Thank you very much. This is a very interesting question. We have thought about it. We were even interested in doing this with functional networks, which is where we would have positive and negative weights. We haven't done it yet, so I have not worked myself directly in the formulation of the geometric normalization. So I'm not sure what will happen. If we are always keeping the maximum weight, then we would delete inhibitory connection. So I don't know if that makes sense. I don't think that makes sense. So we would need to find a way to also keep the inhibition there. I cannot answer directly for now. For now.
Any other question? I will have one if you don't mind, or maybe a couple of questions which are related. So I wonder, I think it's super beautiful, and everybody likes this self-similarity thing. And it's more of a, I wonder how much farther you might have gone. So two questions. One is, have you tried if these results are stable under different renormalization schemes, on the one hand, and then on the other, did you expect or have you tried if this also reflects some self-similarity in dynamics and functional networks? Did you ever try anything like that?
With the first question, no, we didn't try other renormalization techniques. To be honest, in my group they really focus on hyperbolic geometry, and they developed all these tools. So we just wanted to use that model to see if it worked with that. I'm not sure if it would work. I assume if the renormalization is performed like, in a way that keeps self-similarity, then it would work. But we didn't try it. And sorry, the second question was
Functional connectomes or dynamics?
no, that's what I was just telling in the other question. We wanted to explore it, but we didn't have access to functional networks at this amount of scales, and we didn't try it still. I think at some point we will want to do it, but for now we will check it.
Okay, so I think there are no more questions in the room. So thank you very much once again. Next speaker.
And now I sent you an email yesterday.
Let me see. I believe I put it. Cinco minutos. Is your last name? Ah. Something like that. Start from here. Ah, no. since we're almost there, I will introduce the next speaker, who is Michael Merkel, who's going to talk us about autism, specifically redefining autism subtypes via multi-scale brain network topology. Please, the stage is yours.
Thank you very much. So I think I stay here with the microphones. First of all, to the organizers for inviting me to this very interesting and broad conference. You've put together a nice collection of talks, and sorry for sending in the talk a bit late. So I'm currently working at the University of Applied Sciences in Aschaffenburg, which is a suburb of Frankfurt, Germany. Today, I want to move on from what we heard for the healthy brain right now, studying brain connections, to look into a more medical motivated approach. So we start with a clinical motivation on autism. Autism is a disease which many people know. It's usually diagnosed in young children where they have behavioral dysfunctionality, so difficulties to develop language skills, motor problems, or difficulties in building up normal social relationships. The work is based, of course, on a data set. I will introduce you briefly to our methodological framework, which is machine learning. It produces some results which we can discuss, and we want to move on. So the point is to contribute to explainable machine learning and to see if network analysis could help us to understand results which we get for classifying in my type subtypes of autism. This is work in progress, and I'm very happy to take recommendations, comments on what could be interesting here. Let me first give credit to my postdoc, Caroline Alves. She did most of the work in this project, and she will move on to Switzerland next year. So you just register her name, Caroline Alves, going to Geneva from January onwards. Please let me point out some of my collaborators. Francesco Rodriguez from the University of Sao Paulo in Sao Carlos is leading a big group on nonlinear dynamics there, and Patricia Aguaya is working at the Albert Einstein Hospital there, who contributed a medical view on this project. So to start with, this is an illustration of the brain, which I found in one of the very early printed books on the brain by Thomas Willis. So it's on display in the library at the Cathedral Library of Wells, the smallest cathedral city in the UK. And the interesting thing is, the illustrations in this book were made by Christopher Wren, who is quite famous for a very different thing, namely being the architect of London buildings like St. Paul's Cathedral. So in those days, 1680, printed in Geneva, people observed structures of the brain without being able to really explain it. Today, we have much more sophisticated visualization tools. We have fMRI, but the issue is more or less the same, making sense of all the data and of all what we see. There is a medical debate going on right now on how you should diagnose autism. Until quite recently, the Diagnostic and Statistical Manual of Mental Disorder defined 3 subtypes of autism. There was a general definition of autism spectrum disorder, where the focus was more on difficulties in social relationships, communication skills. Another class, restricted motor skills, contributed to the diagnosis, and which probably many of you know, the so-called Asperger syndrome, AS, which is a high functional form of ASD, which comes with very focused interest in certain fields, repetitive behavior, and perhaps some issues in language development. all these classes have not been very sharply defined. And so there was a revision 2 or 3 years ago to combine them into a single diagnosis. And in some sense, we worried a bit. Is this the end of science? You can't differ clearly between 3 sub-cases which come up with, some obvious traits, but there is a lack of, a sharpened view on what could be a good classification. So we think this is a good use case for looking into with new approaches and techniques to see if we could contribute additional evidence on how to make a classification of subtypes in autism. And this is what we want to do in our research here. What is the substantial difference between this Asperger syndrome and general autism manifestations? It has been to some degree seen in clinical observations. We want to do fMRI data analysis of the brain with a focus on explainable AI. So there is, of course, a control group needed. The control group in this case is called typically developed because it's about young children who typically develop according to a certain path. And so TD in this case means healthy control. Keep this in mind. So the data set which we use is publicly available, so you can double check what we do quite easily. It's functional magnetic resonance data of the brain, so blood oxygenation, level-dependent time series data, bold as it has been used many times. We study it for 122 brain regions of interest, and you can download it from this link. It contains about 600 data sets for the control group, so they're typically developed, and about 500 for the various manifestations of autism. It's a bit biased within these manifestations. So for the general autism syndrome, we have 280, and then you see the numbers. This data is represented, visualized using a brain parcellation. The one which we use is slightly different from the one which was on display right now. So we use BASC, the bootstrap analysis with stable clusters, which is implemented in a package. So we just downloaded this package from GitHub and used it as it is. It offers, a variety of different coarse graining levels, and the scale which we went for was the 122 regions of interest of the collected data. So now I would like to show you the framework, how we do the analysis of this fMRI data. So initially, this time series data needs to be sliced. There is a certain windowing technique. For each slice, we form a correlation matrix, and so the connectivity matrix of all those time series results is calculated using the Spearman coefficient. Then we directly work with these connectivity or correlation matrices, which have 122 by 120. So those matrices can be directly used for a machine learning classification approach. So the time series data pulled and transferred to this connectivity matrix is first directly inserted into machine learning routine. The second step, which I will discuss with you afterwards, is how we can use complex network measures and topological data analysis for a second view on the problem. So the first one is, of course, you need to do some optimization. What is the right window size? What are, the best way of doing the correlation of the time series? So to say, the best connectivity matrix. What is the best algorithm? And it comes with a lot of benchmarking. So you run this data time and again through similar setups, and with the objective to obtain the best performance of your total setup, your machine learning. So this took quite a while because it's many iterations. You check for a lot of technical details and what is the right k-fold stratification. And so here you can see that you have, depending on how many data splittings you do, you get different performance of your total framework. So we opted for the typical approach with 10-fold cross-validation. And let me show you some of these performance results. This is a benchmark for different machine learning algorithms. In the first top line, you see CNN, the convolutional neural networks, and then there are a couple of other algorithms for comparison. And so trained on the available data, they all perform pretty well. But there are differences. The CNN performed best, but it's computationally quite expensive. And the support vector machine is doing an almost equal good job, but with a much faster computational effort. So if we study the best available classifier, which we found, you see that there are pretty good results. This is a confusion matrix representation. And so you see the predicted class, the outcome of the machine learning prediction is a certain case, a typical developed or Asperger syndrome or whatever other class. Compared to the actual class. And there is only, a small irritation in the last line, where you see that the two classes, the autism, ASD, and the subclass PDD are in some sense not clearly separated. This is a manifestation of the overall observation that the classification of the subtypes did not work very well. And in this case, it may not be a problem of the machine learning classifier, it may be a problem of the approach, that there is no clear cut label available between those two subtypes. However, there is a pretty clear distinction between those two and the Asperger syndrome, and we didn't see any difficulty with separating this off from the remaining cases of autism. So then we continued with a couple of technical approaches. Recursive feature elimination is a tool to see how many input features are actually needed to achieve this result. Normally, you don't need all what you put in, not all 122 times 122 pieces of information of your correlation matrix. And here you see it saturates after about 100, 120 input features. So there is space for simplifications. You don't need the full information provided in this approach, and this gives rise to making it more explainable. Because if fewer features are sufficient for a similar analysis, you may choose explainable, more understandable approaches. But the first approach was to look into the available representation. We have trained the machine learning model. It performed quite well. So we assume it got something of importance, and we want to see what this is. So we used the Shapley value approach to represent the brain network, the most relevant connections of, which had decisive influence on the outcome of the classification. And I can show you here, how we approach this. We basically found out what were the most relevant features, the most relevant entries in our correlation matrix, and those were selected for the two cases for the Asperger and the remaining things. So we see if we do a, other metrics for these Shapley values, how well those different subcases are separated on the level of the Shapley value representation. So the typically developed first column is pretty clearly separated from the remaining block of the autism cases, and there is a tiny, overlap between the Asperger and the ASD, and so this is in somewhat separated off. You see, and there is only a cosine similarity of about 80%, and the others is pretty much the same, so you can't see any difference between the ASD and the PDD. Now the question is, can we visualize this? And so we basically locate those brain positions on the brain atlas. We pick out what kind of correlations contributed most to the classification success. And we noticed that this is something which you can relate to existing medical knowledge. The most influential correlations were those which affected brain regions known for, typical symptoms in autism diseases. Anterior cerebellum, posterior cerebellum, and so on have all been discussed amply in the medical literature. So here is a list of the most relevant ones. They contain the left orbital frontal region, which has been discussed a lot in medicine, and the prime motor, where the motor control is located, and some temporal pole here in the fourth line, which is relevant for social interaction and language. So in this sense, the machine learning has found what is inconsistency with the existing medical understanding of the brain. And if you visualize it, you can see again the same information on this brain map. So with a focus on the left hemisphere of the brain and a focus on the motor. So the question, are there some, prime motor related aspects are helpful to distinguish between the subcases of autism. So here it's about what is a good region to look at if you want to distinguish between subcases. There are different correlations important for these various cases, but they all make sense with respect to the medical knowledge. So it's all in the paper. If you want to have a closer look, you can see that the ASD, AS, and less so the PDD, because it's not really a separate subcase, and they connect well to what we found in the medical literature. So let me be a bit quick on this and come to our next approach. So now the question is, how could network analysis help to simplify this analysis and to make it a bit more explainable. So far we have used the full connectivity matrix. Now we want to go into a reduced representation. Reduced representation is to understand this connectivity matrix as a graph, as a network, and calculate complex network measures from it. So this comes with a long list of complex network measures. You know probably some of them, between centrality or entropy of degree distribution and whatever. So there is an ample set of such features available which are used to characterize networks. We calculated some 26 or so of those and did a statistical analysis on pairwise interactions. Are they related, to each other? Are they statistically significant on their own to distinguish between subcases? Here you see some are, some are not, and you need a collection of many of those to perform. It's not enough to focus on one of the hemispheres. So in this case, you see they are not statistically significant if you only include network measures drawn from either the left or the right hemisphere. And they are in total not sufficient to achieve the same performance which you can get from the full connectivity matrix. So the performance based on a machine learning classification with complex network measures input is substantially worse. This means there is something missing. The complex network analysis is not sufficient, and what could be missing is higher order interactions of the network. So the natural next step is to say we need to go to topological data analysis on a higher level. And this is what we tried here for the first time in autism, to come up with these Torres RIPS complexes, looking into these various simplex structures, node, edges, triangle, tetrahedron, and so on. So all these lattice structures which may be relevant for distinguishing between those subcases. This is an interesting approach. So we studied it for a number of examples. Fractal dimension provided some significance by its own, but it's a little bit like in the talk we heard yesterday morning. Each individual piece of information is not sufficient for the task. You need to bring it together to look at the collection of elements to achieve, distinguishability between the various results. So here another one, persistence entropy, is good for some kind of separation, but none of them is sufficient for everything you have in mind. And so I'm already running in over time. Let me be a bit quicker. So what we can conclude on this is persistence entropy is a pretty interesting measure. I think it can capture essential differences between ASD and this Asperger subtypes. And for this Asperger, there is a pretty clear case that it comes with a distinct brain connectivity, which we can probe using these topological features. So let me sum up. Our observation here was that the general ASM and PDD classes cannot be distinguished nicely, and there is a good cause for combining them in a single diagnosis. So this is in agreement with the medical community has done. But there is also clear evidence that the Asperger cases form a distinct subset, and that should be maintained. So there are asymmetries in the brain hemisphere connectivities, which are relevant but not sufficient for a classification between those cases. the complex network measures as they were previously are not sufficient to develop predictive power for an ML-based diagnosis alone, but there is promising new directions coming up with topological data analysis. And our next job will be to combine this all into a new classifier and to see how well the topological data plus the complex network measures plus some further understanding may perform on this job. So thank you very much for your patience. I agree that this was a pretty applied talk, but I hope it was in some sense interesting to some of you.
Very interesting indeed, and we have some questions.
Sorry?
What were the age of the brain samples you were showing?
The age?
The age of the brains, the people.
Yes.
Are they adults or are they children?
Mix. I think it's a data set between between 5 and 80, and I have to check out what is the distribution.
The reason is that there's a real opportunity here. I'll speak from personal experience with somebody in my extended family who at age 1 or 2 was diagnosed with pervasive developmental disorder. It was probably a misdiagnosis because his parents ignored it, went through a whole bunch of training exercises. He's now an adult, and it's clear that he has something like Asperger's syndrome. He's very, he has a perfectly fine job. He's married. He's socially awkward, but so if there had been a test along the lines of what you're proposing at the level of a child, of an infant, a toddler, whatever, it could have a very large impact, at least based on my personal family experience.
I agree, and this is completely our motivation, because there have been so many misdiagnosed cases. The medical community abolished subtyping. Well, this is one approach of doing it. Another one is to say, could we contribute to a better distinction between those subcases to give an early indication that it's Asperger, that it's a high functionality form of autism, and that you can make a nice and decent life by arranging yourself with this condition which you have. And I think it would be particularly helpful for those affected by Asperger.
Time for a last question over there.
Yeah, thanks for the excellent talk. It's more or less in the same sense. Actually, just to detail a little bit, have you tested then the same technique but using data sets like focus on different ages, like to see if there's any difference in the behavior or how better they perform depending on that's data set, or that's something that's actually very, I don't know, something that you can investigate because you can find data sets in that way? Or...
So unfortunately, it's not very easy to find high quality data sets on this thing. Because fMRI is a pretty expensive technique. This was already a collaboration of 10 major US university hospitals putting together their data in a publicly available data set. There are not too many of those. If you have one in mind, which we could test, please approach me. This is very interesting for us because the generalizability of this approach depends heavily on the data you use. And it would be very interesting to see if the generalizability improves using complex network analysis. If details of the correlation matrix, which may be specific to some of these data sets, could be in some sense avoided by going to a more network-based approach.
I see.
Okay, thank you very much. So thank the speaker again, please. And now we have coffee break. I don't know if you're gonna make it shorter because we are a little bit late with the talks or no. Okay, so we have a regular coffee break. Enjoy it.